Hi, this presentation or this video is about how to find primers using the National Center for Biotechnology Information, NCBI, and their web pages. This is the leading place if you want to find any DNA sequence. And the first step when you want to design primers is to find some DNA. This is the opening page. A couple of words about the page as such. To the left is a resource list. I'll get back to that in a second. And the right is popular resources, it states. And that's because that's the most used of the many resources you can find in NCBI. In top you see PubMed, which is one of the best, in my opinion, best places to search for scientific-like literature within science, mostly biology, molecular biology, biomedicine, but also pretty good covered in chemistry and even hardcore physics. PubMed is the way to go there. Blast is to search for homology with DNA sequences. Nucleotide is nucleotide sequences. You can find genomes. You can find single nucleotide polymorphism. You can find gene proteins. And chemistry, out of my field. On the other side, you can find different resources within the different subjects. My recommendation is click around, see what you can find. It's without the scope of this presentation to go through it all. You can also have a look in the bottom, find basically the same stuff. Important when you want to search is to narrow your search. And in one way it's quite easy, and another way it's pretty hard. It's pretty hard because you can see there are a number of ways you can search. And there is a huge amount of different databases, literature databases, etc. you can search. And a lot of it just has a pretty, it has an abbreviation which really doesn't tell anything but the nerds what it is all about. So, for this case, I will search for a gene. So I will select the gene database. Little about the things you can do here is, again, resources. As you can see on the resource list, here you just have it with a figure, but you can basically find the same thing. Submit is when you want to submit, and as I said, it is the database for DNA sequences. All things sequenced. Maybe there are some things sequenced in North Korea or Iran or somewhere obscure where they keep things hidden from the rest of the world. But apart from that, anything else should be in the GenBank, as it's called. It's a database with all the genetic information, with all the DNA sequences. I selected I want to look for a gene, and we will stay in microbiology. You got a project to solve, but I would cover something else or show you an example of something else so you have the chance to try your own stuff yourself. I would like to work with lactobacillus, and I would like to work with a single copy gene in lactobacillus called TUF. It's a elongation factor. And let's see what happens. This is the first step when you want to design primers. It is to get in there to find the sequence in your area you want to work with and really try to narrow it down with whatever it is. You can see I get a number of choices. And lactobacillus is a very common bacteria, and it's, as you can probably know, used for a lot of different things. Acidophilus is used for making both cheese and yogurt and stuff like that. We also got Lactobacillus sakei. You should know a lot about the Japanese kitchen, and especially what you can drink in Japan, before you know what Lactobacillus sakei is involved in. So a number of possibilities. And I actually looked for something in, because I would like to see it from Lactobacillus casei. So I just have to go through the list. And it doesn't show up. Fine. We take a cousin. It's called Brevis. It's still the elongation factor TU. Short is TUF. You can click on it. If you're not happy with what you find, or you find multiple, you have to narrow your search. And the easiest way is simply to add more things up here. In your project, you're going to work with genes, you're going to work with an organism. So organism name and the name of whatever gene you're going to work with. is a good point to start. It shouldn't make a difference if you choose the gene database or the nucleotide database. It should give pretty similar results. It is narrowing. You narrow things down if you go for the gene database instead of the nucleotide database, but it still can give you a lot. Anyway, let's click on that. We get into the next webpage and we get a lot of information. Here we have the tough gene. It says something about markers and repeat regions. It's not there. It's primarily something you see in eukaryotes. This is a prokaryote. We don't have a SNP map the same way in prokaryotes as we have in eukaryotes. So don't worry about that. You can go to reference sequence details. All sequences are, held up against a reference database, which is the sequence. A lot of things have differences depending on where things are isolated. Even if it goes to humans, we have a lot of SNPs. So we have a reference sequence, and that's the sequence, and then we compare things to that. A reference sequences might be a good place to start. You can see, you can try to click it, but the problem is when you go for that, as you can see, if we follow my cursor, you get the whole complete genome. And what you see here is a number of the different genes in that gene. We don't want to do that. We just want to work with the tough gene from Lactobacillus brachius. So I can see the nucleotides graphically. We get into more detail, pretty much the same picture as we saw before. We could also go to GenBank, and that's usually where I go, but hey, come on, it's up to you. So here you see a number of things. We start from the top. Authors. It's basically the people who was involved in the work making that sequence. You see there are a number of authors, and they have made comparative genomics of lactic acid bacteria. You can find the paper where they published. It's quite a while ago, and it's the consortium was who did the sequencing title. It's a direct submission. Sometimes GenBank has a number of people employed, and they will pick up sequences if they have not been submitted from publications, and a lot of other stuff. And source, base number 1 to 1549. Lactobacillus brachius, ATCC, is American Tissue and Cell Culture Collection. It's a big bank of different cell cultures and strains of bacteria, and you can actually mail them and order this strain. So if you want to do, let's say, repeat this experiment, or you want to work with this particular bacteria, you can order this from ATCC. And this is one of the big ones. There's also a big one in Germany, Deutsche Tissue and Zellkultur, I can't remember the name, but they cover pretty much the same thing. And we can order sequences there. A lot of other things. The gene starts at base number 179 up to 1369. So we have a little more sequence than we actually covers the gene. CDS, coding sequence, and it's a tough gene. Here we have the amino acid sequence. You can find the protein using the protein ID. This is the gene ID. And down here we have the sequence from 1 to 1549. And our gene, the tough gene, is then located somewhere in here. Excellent. So now what? Well, you're going to design primers. So let's go and pick the primers. This is a number, and that's one way to do it. Or you can upload the FASTA file or the FASTA file. Depends who you are. Americans say FASTA. A lot of the rest of the world says FASTA. Doesn't really matter. Danes say FASTA. But anyway, that's another way to show the sequence. 60 bases in a line. If you remember what it said in GenBank, it said we started at 179 and ended at 1369. So one way to do things is to go to FASTA. 60 characters in each line. That means the first 3 lines are outside the gene, and then it's 1549, and we stopped at 1369. So again, subtract something like 180. So if we just select this sequence and copy, I didn't do it correctly, just sorry. So here we are, and copy, and pick primers, and just paste the file, the sequence. That's it. And there's some numbers here. Get rid of that. So now we got the sequence in there. In there we want to have some primers. If you have already worked with one primer that worked fantastically and you just need the other one, you can pop the sequence in. The next thing you can do is PCR product size and the number of primers to return. The product size, you're going to use this for qPCR a little later. It started at 17 base pairs, and that's actually quite alright, because 70 base pairs are twice the size of primer dimers. So that's good. You were able to see the product on a gel and be sure it's a product and not just primer dimers. So we keep the 70. For qPCR, 1000 base pairs is usually way too much. Let's go for 250. Numbers of primers to return, 10. It's a bit too much for your project. You're only allowed to order 1 or 2 sets of primers. That means 2 or 4 primers. But if you are in a professional surroundings, in a hospital or in a research lab, 10 and ordering all of them at the same time is a good idea, because you can optimize the different primer sets and find the best one. Save a lot of time, save a lot of money, instead of ordering 1 or 2, check it out, order a new one. Primers are cheap. Your time and all the other reagents are the expensive thing. Ordering 10 in normal circumstances, whoops, I forgot to move the cursor, is a good idea. So we say 5. And I got a 1 up here, I got a delete. Okay, primer melting temperatures between 57 and 63. It's okay. We don't want more than 3 degrees difference between the 2 primers. Exon entrant selection is something you do when you want to work with gene expression assays in your carriers. We don't do that. Primer pair specifically checking parameters, automatic, database, RefSeq, messenger RNA. It's a good place to start because it is a gene. You could also, if you're not working with a gene, go for genomes or genomes of selected organisms. But let's just start with it. And what it will do is it will take the sequence for the primers and see if they fit anything else. It's bound to do, but the important thing is, does it fit anything in Lactobacillus brevis? But that's the BLAST part of the primer design project. BLAST is a search for homology in the database. So is there anything, anywhere these primers will fit apart from what we want to design it for? And that's an important thing to know, because this could give you problems. So it will check that out. Organism, this is actually the number for Lactobacillus brevis, so we are happy. We can always put in more organisms. Max target size, yeah, I don't really know what it is. You can get some information, and you see it tells us the max size that your primers can generate, PCR product they can generate, and 4,000 base pairs. Well, if it wouldn't really be a problem. You could put this number lower, and then you would probably not get a lot of hits. But anyway, allow primer to amplify messenger RNA splice variants, something you see in eukaryotes. This is a prokaryote, so no worry. Advanced parameters, a lot of information, and it's really for the nerds to go in here. You can check for the GC content. You can do a lot of things. And if you've got a job where your job is to design primers day in, day out, be more than welcome. Test it out. But for the rest of us, mortal souls, stay with this. Click the Get Primers. Primer specific sequences for the selected are not present. Okay, I chose the wrong database. I was wondering about that. We go for representative genomes instead. It took a while. That's why I clicked around. I'm sitting here in the afternoon. If you look at my computer, it's around 6 o'clock. 6 hours from 6 o'clock is 12 o'clock East Coast USA, and 9 hours less than that, that's 9 o'clock in the morning West Coast USA. 80% of all people designing primers in the world sits in the other US. So a good idea is to do this when you're in Denmark. Do it in the morning, because as you can see, it's right now doing the job. Tell us, yes, that's fine. what it tells us there was that the sequence resembled Lactobacillus brevis, and that's fine. Again, it will take a while, and the next presentation I will do is about how data is presented. So this was the first video about primer design.